Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the muscle contraction obviously, and this is the second part of the muscle contraction. And in the first part I already explained and uploaded that video in my channel. So this is the second part of the muscle contraction. Okay, so let's continue. The coupling of actin, myosin and ATP hydrolysis, and this is the rough diagram or rough sketch, you can say, just for the explanation of this process. So what is saying that myosin head without ATP locks tightly onto the actin filament. Clear? So myosin head, this is tightly packed or tightly bind with this actin filament when without ATP. Clear? Without ATP, it can easily bind or it remain bound with this actin filament. Okay, this is the actin filament, and this is myosin head. Okay. So what is happening when ATP just enter? They bind with this myosin head. Okay, when ATP binds with this myosin head, then what will happen? It change the conformation of the actin binding site. So here the binding, that is lock the myosin head with the actin, this become totally stop. That means why? Because ATP bind with this myosin head and the conformation of this binding site, that becomes change, and that's why the myosin head never bind with this actin filament in presence of ATP. It dislodge from this actin filament. Clear? So after dislodging, what happened? This reduces the affinity of the head for actin, and to move along the filament it now tries to move along the filament. Okay, it tries to move along the filament, and this ATP, when the ATP hydrolysis is occurring, then form ADP and PI. Okay, sorry. Okay, so what is happening? That when ATP hydrolysis is occurring, then ADP and PI form, and when ADP and PI form, at that moment also ADP and PI that tightly bound with this myosin head, and you can see here ATP hydrolysis is occurring and ADP PI form that is still bound with this myosin head. Okay, and when this PI release from this part or myosin head, then what will happen? That binding of the myosin head to the new site on the actin filament is occurring. That means what is saying that if exactly 1 second. I just have to or just try to understand you that the place is changed here. The place where it was first there, now it was not there. Okay. Suppose these 3 dots are there. So in the second situation, so second part is that means in the second part, this is the third part, you can say third part. Okay, so just I have to try to draw or to understand you that the situation is changing or the place is changing here. Okay, in the first situation it was just bound with the second point. In the last situation it was just bound with the third point. Just I want to try to understand you that the position is changed here. Okay, so what when this ADP and PI form, and by the hydrolysis process of ATP, then it was bound with this myosin head. But when PI released, then myosin head has or arise a chance of the myosin head to bind with the actin filament or another position of the actin filament. Again I repeat this one, that when ATP hydrolysis completed, then ADP and PI form, and then still myosin head there, ADP and PI already present. So myosin head had no chance to bind with the any position of the actin filament. But when the PI is released from this position, then myosin head tend to bind another place of the actin filament. Okay, so in the last situation, you can see that ADP also released, and when ADP also released, then it is tightly bound with the actin filament, and here the position obviously changed. And you have a question that why the position is changed, because you first here you have already seen that the conformation is already changed, where the site of the binding site of the actin and the myosin head. Okay. So if the myosin head tries to bind with this actin filament this position, it was impossible for this myosin head to bind with that position. Why? Because conformational change is occurring at the binding site. That's why myosin head had no any advantage that it can bind with the same position of the actin filament. Clear? So position is changed here. Release triggers the power stroke, and the losses its bound ADP and returns to the start of a new cycle. So what is saying that new site on the actin filament, the myosin head binds, and this so this release triggers the power stroke and loses its bound ADP and returns to the start of a new cycle. So here a power stroke is formed. How the power stroke is formed? That the releasing the binding site from the actin, releasing of myosin head from the binding site of the actin filament, and again bind with the another position of the actin filament, and here a power stroke is just formed. Okay. Next is that the smooth muscle contraction. This muscle contraction is related with the skeletal muscle, and here this is the smooth muscle contraction. Smooth muscle contraction is very easy, that here the contraction of the smooth muscle regulated by primarily a pathway in which the myosin regulatory light chain is associated with the myosin 2. Undergoes phosphorylation and dephosphorylation. Here also phosphorylation and dephosphorylation procedure is occurring, but this total muscle contraction is dependent on the enzyme that is called the myosin light chain kinase. This enzyme is very important because the phosphorylation of this enzyme that helps in the contraction of the muscle, or it can regulate the contraction of the muscle, and this enzyme is activated by the calcium. If the calcium level is high, where in the cytosolic area or cytoplasm or cytosol you can say, so here the calcium level is high, that means this calcium level actually helps in the phosphorylation process of the myosin light chain kinase. Clear? These enzymes. And here calcium, that is very important. Why? It is another way dependent on the another protein. This protein is called the calmodulin protein. This is the calcium binding protein. So what is happening here? This calcium binds with the calmodulin and form a complex which binds to the myosin, and this myosin, that is called the thick filament, you can say the thick filament regulation. Sometimes, so in that way this process is occurring. So first calcium level that becomes very high, and this help in the phosphorylation process of this enzyme, and this enzyme also regulate the myosin 2 or light, that is associated with the light chain. And another important thing is this is the calcium dependent pathway. Another thing that is the calcium independent pathway. In this situation, the muscle contraction is regulated by another protein. This is the rho kinase protein, and this activates this regulatory light chain by the phosphorylation. So phosphorylation, dephosphorylation, these are the happening here. These are the main important thing you can say, but these are controlled by another way here. One in the calcium dependent pathway and another is the calcium independent pathway, but two are controlled by the two enzymes. In the first case, the enzyme is that myosin light chain kinase. In the second case, the enzyme is that rho kinase. So this is the smooth muscle contraction process. So muscle contraction process, this is the last part of the muscle contraction process, and total muscle contraction and actin filament structure also I explained and uploaded in my channel, so you just check out that all. Okay, so thank you.